Hi everyone, welcome back to the channel. Be sure to subscribe our channel by clicking on the subscribe button. Today I'm going to cover chapter 5, structure function of macromolecules or large biological molecules. So macromolecules are polymers built from monomers. The carbohydrates is an example, serve as a fuel and building materials. Lipids are a diverse group of hydrophobic molecules. Proteins include a diversity of structure, resulting in a wide range of functions. Nucleic acids store, transmit, and help express hereditary information. So let's get started. So we start with the carbohydrates. But first, let's take a look at the monomers versus polymers. So as you see here in this figure, polymers are made of repeating units of building blocks called monomers, and there are two processes. Dehydration reaction is the synthesizing of polymer, and hydrolysis is breaking down the polymer to the monomers. Okay, our first macromolecule is carbohydrates. They are the fuel and building material. Monosaccharide is their monomers, which is CH2O molecular formula. The glucose is the most common monosaccharide found in many places, including blood. And again, there are other monosaccharides, including glyceraldehyde, including ribose, ribulose, glucose, galactose, and fructose. Disaccharides are actually the two monosaccharides. They are actually covalently bonded at a special bond called glycosidic linkage and make actually disaccharides like sucrose. Polysaccharides are several monomers, polymers of the sugars that they are used storage and structure roles. Best example is starch in plants, and basically it's a storage polysaccharides of plants that's commonly used, which is made of amylose, and glycogen in animals, which is a storage polysaccharide of animals stored mainly in liver and muscle cells. Okay. And the cellulose is another polysaccharide for plants and found in plant cell walls. So our second group is lipids, which is basically a large group of hydrophobic molecules that they are not really true polymers. They are made of smaller pieces, but they are not really true polymers. For example, fats, they are constructed from two types of molecules, glycerol plus fatty acids. You can see here the glycerol alcohol, which is covalently bonded to three fatty acids tails here. Okay. Fats can be saturated or unsaturated. Saturated fats have the maximum number of hydrogen atoms, possibly in their double bonds. Examples: animal fats. And unsaturated fatty acids are the ones that actually they have one or more double bonds, so they are not saturated. Examples are plant fats, fish fats. They are liquid, usually in room temperature. So diet rich in the saturated fats may contribute to cardiovascular diseases through plaques deposits in the blood vessels. Okay. Omega-3 fatty acids, which are required for normal growth, and they are thought to provide protection against cardiovascular disease. Phospholipids are another group in these lipids that they have two fatty acids and one phosphate group head here. You can see here. Steroids are a group that actually this carbon skeleton with the four fused rings you see here. Cholesterol is an example of the steroid. Proteins is a very large group of more than 1 million proteins, and it's very unique because actually they are used actually in so many places, including these 6 functions like enzymes, storage, defensive, transport, hormonal, contractile, motor proteins, receptor, and structural proteins. So they are they are also polypeptides. They are basically made of 20 amino acids that we know. Okay, So these amino acids have R groups, and they have amino group and carboxylic group. So polypeptide is basically the amino acids linked by covalent bonds, a special bond here named peptide bonds. Proteins have also something that other groups don't have, which is their 3D structure and then their configuration. Their structure can be primary, secondary, tertiary, quaternary. You can see in this picture. Primary is their open structure, and secondary it has the hydrogen bonds. It can be alpha helix, beta sheets, beta plated sheets. Tertiary, more folded, and the quaternary is several, more than one tertiary got together. So in secondary structure, we also see hydrogen bonds, and we see disulfide bridges and ionic bonds. Quaternary structure is their final structure. You see here some proteins like hemoglobin. You see here it's common in our red blood cells. Sickle cell disease is there is only one change in the primary structure of the protein. You can see in number 6 position, glutamine replaced with valine, and actually that causes sickle cell. What determines protein structure? Protein structure actually can be changed, something called denaturation by high temperatures, pH, salt, again, concentrations. There is a group of proteins called chaperones. Chaperones, they actually help the folding of the proteins. There are a number of areas that studied in proteins, including X-ray crystallography, determining protein structure, NMR, spectroscopy, and bioinformatics, basically some techniques that use in protein studies. Nucleic acid is the last group. As you see, we have DNA and RNA, deoxyribonucleic acids and ribonucleic acids, two major nucleic acids. They make the genes, and you can see here nucleic acid actually made of nucleosides, and basically that's nitrogenous base and the sugars. And so the backbones run in the opposite 5' to 3' directions, and the complementary pairing, A always with T and G always with the C. And this concludes our video on chapter 5, protein structure and function of large molecules or macromolecules. See you in our next video.